Very hearty good afternoon to all the speakers from national research and development institutions, state central universities of India, chairman, vice chairman, directors of the board, principal, faculties of various disciplines, and all participant students from India and other parts. The e-conference for 10 days is being organized by the GERMS Arts and Science College, affiliated to University of Calicut, is one of the best college having distinction at all its level. I could see highly unique and talented speakers around the country, namely Professor R.S. Verma from IIT Chennai, Professor Arun Lahiri Majumdar, INSA Senior Professor, Bose Institute, Calcutta, Professor Pranab Goswami, Indian Institute of Technology, Guwahati, Professor D. Girija, Kerala Agriculture University, Dr. Mallikarjuna Swamy from Senior Scientist from KFRI, Professor Abhay Pandey, Central University, Allahabad, Professor Bhattacharya, Assistant Director, ICMR, Regional Subunit from Bhubaneswar, Dr. Prasenjit Manna, Ramalinga Swamy, and he is also a Principal Scientist of CSIR NIST, Jorhat and the DST, Dr. Bula Chowdhury, Senior Scientist from Guwahati Biotech Park, and Dr. Sanjay Banerjee, National Institute of Pharmaceutical Education and Research, Guwahati, will be delivering lectures from today till 23rd September on topics very much relevant to the theme. I'm sure and confident that all the participants is going to have excellent opportunities to listen and interact with the learned speakers during the interactive session every day. Life sciences research and development has opened new challenges and opportunities. If I speak about my times of research and MSc, graduate or postgraduate, there was hardly any information about PCR. Just for an example, why I'm mentioning PCR, because nowadays a layman, even a rickshawala knows what is PCR technique, just because of the COVID. But you know, as a student of MSc, just started its research. And if I see the developments in research for the past, structure of DNA, that time, during 1960s and 70s, Watson and Crick, we used to learn a lot from different books. Then coming to the restriction endonuclease and genetic engineering that came in 1978, and DNA sequencing from 1984. Coming to that with the microbiome, that is with the recent developments it started. This is one of the newest and potentially most interesting areas in biopharma these days. And biospace regularly covers news of companies working in this area. In short, our bodies play host to a broad arrays of microorganisms. Interestingly, research is showing that makeup of these populations play a significant role in our health. In fact, number of resource persons what we have today and during the seminar are going to touch many recent aspects in this area. Then coming to the stem cell research breakthrough and discoveries, I'm very happy to say that Dr. Professor R.S. Verma from IIT Chennai, he has got an authority in stem cell research. Fortunately, it came in 2012, the most recent developments, but he is going to talk on the stem cell research. That will be another opportunity for the students to listen the most research developments in the stem cell research. Beyond the pale, experts in biopharma tend to be focused on developing the next small molecules of biological therapeutic, but a number of companies are looking beyond the pill to digital health products and wearable devices. Personalized medicine, that's another area. This is briefly following enough about each patient's genome to deliver drugs that are most effective for the individual. This is particularly being approached in the oncology arena. Increasingly, cancer drugs are focused on subpopulations of cancer, usually ones that are recognized by specific biomarkers. All these areas will be touched during these 10 days from today onwards. The gene therapy. For years, gene therapy was something of the holy grails of research. And in recent years, it has become a really, despite the efforts, only a few gene therapies have made it to the market. Artificial intelligence and machine learning, this is one of the area even now nowadays is working with a lot of people working on these areas. Biopharma and biotech companies are increasingly looking to data science, artificial intelligence, and machine learning to give new insights into drug development. And they have not only the ones developed in the health oncology cloud platform, which includes an electronic medical record system for oncology, advanced analytics, patient portal, and integrated billing. The globalization of scientific and technical expertise also means that many scientists and other individuals around the world are generating breakthroughs in the life sciences and related technologies. Globalization, biosecurity, and the future of life sciences examines the current trends and future objectives of research in public health, life sciences, and biomedical research area. Now, some of the recent current research and the challenges what we have it now today. Various types of diseases and their control. That is what currently the people are rating as an example of COVID-19. Food security. These are some of the areas now people are looking and develop number of devices and the techniques. Understanding of the brain, endocrine system, conservation of nature, and climate change. Considering all these important developments in the sciences, our college is organizing a e-conference for 10 days, and the speeches are to be delivered by the speakers of national institutions, universities representing all the parts of the country. All the lectures selected by the speakers of their research of the area of research will be highly informative and useful to the faculties and students. I convey my greatest appreciation and very sincere thanks to all the speakers from different parts of India for their concern to be the part of the conference, organized jointly by the faculties of microbiology and biotechnology departments of Germs Arts and Science College. I also convey my thanks to the entire team of the organizing committee of the e-conference for their continuous, untiring efforts for the successful coordination of the e-conference. And I hope the same type of conference will continue in future too. I'm extremely thankful to the chairman and the management of the college for their excellent support, as usual, for all the time. Thank you very much for giving me an opportunity to become the part of one of the very important event that we are going to have from today till 23rd. Thank you very much.
Thank you, sir. It was a very heartwarming speech by you, and it was very inspiring also. Now, we move on to the main event of our today's session. I request our Honorable Chairman, Shri Manjalanguri Ali sir, for the inaugural address.
Thank you very much. Respected Research Director of the College, Dr. B.G. Unni, Principal Dr. Naveen Mohan, academicians and research scholars from different universities and other reputed institutions, and my dear friends. First of all, I wish to express my gratitude for inviting me for the inauguration of the 10-day conference. 10-day e-conference, in fact, on current trends in life sciences, organized by Microbiology and Biotechnology of the Department of Jeffs College. I think this e-conference is the first of this type under Calicut University, and the organizing departments have done a good job in this phase. With the guidance of Research Director and our Principal, Dr. Naveen. On this occasion, I wish to mention that both microbiology and biotechnology department of this college are among the best departments under the university. And this year, our biotechnology department achieved 2 ranks, and microbiology department achieved 3rd rank in the final year degree result. On this occasion, I wish to congratulate the academic team of the department for good achievement. The topics for the 10 days conference, such as COVID scenario, environmental issues, waste management, plant diseases, entrepreneurship, represent pharmaceutical researchers, are very relevant, and the participation from different walks of life is itself is evidence of the academic interest. I had some experience of the issue of the waste management as the Minister of Urban Affairs of the state government during the last ministry. I am very proud to say that Clean Kerala company, the company started for the e-waste and plastic waste management of the state, was one of my contributions to the state as a minister. And that was the second in India, then too. However, I know that the problems related with the waste management still persist as one of the unsolved questions for every government. Hence, serious studies in these directions can be conducted by the departments of our universities. I am sure that this conference will be a platform for serious discussions with all, which will be useful for entire society. On this occasion, I would like to mention about the mega project of our geology department. The department has undertaken a project about the reason and solutions of the landslides in Kerala, which occurs every every year, with special reference to the Malappuram district and Wayanad district in collaboration with different universities and institutions. I once again congratulate all of you for organizing such a conference, and both the permission of all of you, I officially inaugurate this 10-day national e-conference. Thank you all. Thank you very much. Thanks. What happened, Dr. Naveen?
So there is some connection problem, I hope. Dr. Finos, thank you, sir. Thank you, sir. Thank you, Chairman, sir, for the speech and for inaugurating.
Okay, thank you.
Thank you, sir.
Thank you. Thank you very much. Okay.
Yes, sir. I think Dr. Finos, some problem with the network. Dr. Finos, are you there?
I think you go with the next programs.
Yes.
Next items, I think the Nawaz is going to speak. That's what I was told.
No, sir. It's about introducing the speakers.
Yes.
Yes, sir.
Yes. You can go ahead with that.
Yes, sir. So we have with us Professor Abhay Kumar Pandey, who is a professor and former head of biochemistry from Allahabad Central University. So he'll be engaging a technical session today on oxidative stress and associated complications. And we do have the presence of Dr. Bula Chowdhury, senior scientist, Guwahati Biotech Park, IIT Guwahati, Assam. And we do have the presence of Dr. Prasenjit Manna, his Ramalingam Swami Fellow and senior scientist of CSIR NIST. And welcome you, sir. And I'll just introduce the other technical session engaged by other faculties or other scientists. We have Professor D. Girija, Professor Agricultural Microbiology, and she's also registrar for Kerala Agricultural University. Yeah, Dr. Finos?
Yeah, yes, sir. Yes, sir. Can you hear me?
Yeah, yeah. Can you just continue?
Okay, sure, sir. Sure, sir.
Yeah. So I was reading out Professor D. Girija.
Okay, sure, sir. And so I apologize. Yeah, I'm having some network issues. Sorry. Okay, so let's go on with the program. So it's my duty to welcome all the guests and to introduce all the guests to the eminent participants who are present over here. So I like to take you through just a small presentation, so it will be better for you to understand all of them. Okay.
Yes. Go ahead.
Navin sir, is it okay? Is it
Yeah,
audible?
fine.
Yeah,
Is
fine.
it visible?
Yes, yes. Go ahead.
Okay. So we are presently conducting 10 days seminar on national conference. Okay. So our main topic is current trends in life sciences. Here we are dealing with all the recent topics and present COVID, everything. We are dealing in short everything. So the main duty for me today is to introduce the speakers to all the participants present over here. So first of all, we'll be having the first session dealt by Professor Abhay Kumar Pandey sir. He is present along with us, and he is the professor of biochemistry, and he is also former head and chairman, Department of Chemistry, University of Allahabad. He completed his PhD in biochemistry in 1993 from University of Allahabad, and he has more than 90 publications to his credits and many research articles in one of the premier research journals published by Springer, elsewhere, CRC Plus, and other publishers. And he has received a number of honors. To name a few, Dr. Sarvepalli Radhakrishnan Education Promotion Award 2019 conferred by GES in New Delhi. He is a Distinguished Scientist Award 2016 conferred by SPR, then Distinguished Academician Honor in 2012, and Fellow Award 2011 to 12 conferred by Society for Plant Research. And he has a total experience of 25 years in graduate and postgraduate teaching as well as in research. And his main research topics are biochemistry, redox biology, toxicology, ethnopharmacology, and cancer biology. Our next speaker is Professor D. Girija. She's a professor as well as the registrar of Kerala Agriculture University, and she's presently working in the Agriculture Microbiology Department. And she has a number of inventions to her credit, and she has developed efficient method for conversion of human hair waste into liquid fertilizer, and she has isolated and formulated commercial composting tonic for efficient decomposition of biosolid waste using various microbes. She has also designed KEU Smart BioIn, and it is used for composting household biodegradable waste. Then she has also isolated and characterized novel nitrogen-fixing actinobacteria from the rhizosphere soils of black pepper from Wayanad regions. Then she has metagenomic analysis of bacterial diversity from Pokali rice ecosystem. And she is currently handling a number of projects. She is heading the All India Network project on soil biodiversity and fertilizers. Then Center of Excellence in Microbiology Technology is one of her achievements. And she has isolated native actinobacteria for exploitation in biosolid waste management and also utilization of human hair as a fertilizer. And she has won various best paper and poster awards, and she has won Young Scientist Award in 2000 instituted by the State Committee on Science, Technology and Environment in Kerala Science Congress. Our third speaker is Dr. G.E. Mallikarjuna Swamy. Presently, he is working as a senior scientist, Department of Forest Pathology, KFRI, Thrissur. And presently, he is heading the scientist in charge at KFRI Subcenter, Kerala Forest Research Institute, Nilambur. He has specialized in mycology and forest pathology. He has a PhD in applied botany and is specialized in forest pathology from Kuvempu University, Karnataka. And his main research interests are mycology, plant pathology, medicinal plant pathology, microbial diversity, bioremediation of pesticides, conservation of microbes, building up of database of fungi, ecosystem services, and urban green space development. And he is also involved in biological management of forest nurseries diseases, pre and post infection changes in phytochemical constituents of medicinal plants, soil microflora, LTM of forest ecosystem for microbial dynamics. He has established a live microbial culture collection from different forest ecosystems of Kerala, and he has published more than 35 research articles. Next, Professor Pranab Goswami. He's a professor for biosensors and biofuels, and he is from IIT Guwahati. He completed his PhD degree in 1994 in the area of biotechnology. He was a Boycast Fellow of DST at the University of Massachusetts, Boston. He served as heads in the Department of Biosciences and Bioengineering at IIT Guwahati. His primary research area is biosensors, and his main topic is with an emphasis on the development of novel biorecognition systems and signal transduction platforms for malaria, myocardial infarctions, and alcohol. And his group has many patents to his credit. And he has developed many novel aptamers as recognition molecules for the detection and diagnosis of malaria and myocardial infarction. Presently, he has a proof of concept developed in his lab and has already been translated into portable kits. And this has been used for the detection of malaria, alcohol, methanol, and formaldehyde recently. And two of these kits have been dedicated to the nation by the Honorable HRD Minister of India. Then he has published more than 100 peer-reviewed scientific research papers, and presently he has filed 11 patent applications. He also served as a visiting professor at the University of Alberta, Canada. Our next speaker is Professor R.S. Verma. Presently, he is working as a professor, Stem Cell and Molecular Biology Laboratory, Bhupat and Jyoti Mehta School of Biosciences, Department of Biotechnology, IIT Chennai. He completed his PhD from JNU, New Delhi in 1984, and his broad area was stem cell and molecular biology. And he has been handling immunology, biochemistry, biotechnology, microbiology, stem cell research papers for the last 16 years. He has a lot of honors to his credit. He has achieved Lifetime Achievement Award from Natural Science Foundation, Tamil Nadu in 2017. He is a member Board of Governor, National Institute of Pharmaceutical Education and Research, Mohali, Chandigarh, Fellow for the Biotech Research Society of India, BRSI, and he's also a Fellow of National Academy of Science, Allahabad. And his main research interests are stem cell regeneration for tissue engineering, stem cell scaffolds for the cardiac, liver, tissue generation. And he has also interest in cancer stem cell and their targeted therapeutics, biomarker and pathway studies in Fanconi anemia, and also construction of production of bacterial and humanized immunotoxins for cancer therapeutics. He's also interested in monoclonal antibody production as well as cancer drug delivery. Next speaker is Dr. Devdatta Bhattacharya. He is presently acting as the Scientist D, Assistant Director of Microbiology, ICMR RMRC Orissa. And he's a recipient of ICMR Centenary Gold Medal. Then he's a member of Emergency Response Team, deputed for management of the outbreak of various infectious diseases by Minister of Health and Family Welfare, Government of India. And he has been involved in the National Rotavirus Surveillance Research Project, and is currently handling 3 projects as principal investigator, which is mainly funded by ICMR and DBT. And he has also published 40 scientific papers on antibiotic resistant mechanism, diarrhea, cholera, hepatitis B, dengue, swine flu, chikungunya, rotavirus, Wolbachia in national and international journals. He has also developed the first policy brief of anthrax control in India. He is the nodal officer of Media and Communication Cell of RMRC Bhubaneswar. He is currently the in charge of high throughput machine for SARS-CoV-2 diagnosis and serodiagnosis of COVID-19 at ICMR RMRC Bhubaneswar. Next is Dr. Bula Chaudhary. She is a senior scientist, Guwahati Biotech Park, IIT Guwahati, Assam. She completed her PhD in biotechnology from Guwahati University, Assam in 2008. And she is having administrative research as well as teaching experience for the past 20 years. She is working as the coordinating officer. It is an honorary position for the Northeastern Cell for Regional Development Corporation. It is a DSIR institute, which is Department of Scientific and Industrial Research, Government of India since 2017. Her research interests are molecular biotechnology, genetic transformation, antimicrobial activity studies, confocal and fluorescent microscopy, spectroscopy, fermentation facilities, etc. And she has a lot of research papers, review articles, and books, papers, edited articles, conference proceedings, seminars, and entrepreneurship coordinated developments, then training programs coordinated on different microscopic techniques, as well as microbial and biology techniques. She has been conducting a internship program, and presently she has completed 19 batches. A popular lecture is the essential of entrepreneurship development for commercialization. Next speaker is Dr. Prasenjit Manna. He's along with us. Then he is a Ramalinga Swamy Fellow as well as a Senior Scientist for CSIR Northeastern Institute of Science and Technology, Jorhat, Assam. He completed his Doctor of Philosophy in 2010 in Molecular Medicine from Bose Institute, Kolkata, West Bengal. He has done pre-doctoral teaching assistant post in Bose Institute, India. He has been holding postdoctoral teaching assistant position in LSU Health Sciences Center, USA. He has published 64 articles in peer-reviewed journals. He's a reviewer of more than 20 peer-reviewed journals, and to name a few, it is only BMC Pharmacology and Molecular Biology reports. These are all highly ranked research journals. He has long experience in isolation and characterization of bioactive molecules, especially proteins, small organic molecules from the traditional Indian medicinal plants. And his research interest is to discover a relatively low-cost dietary supplement that is especially vitamins, micronutrients, etc., that could be used as an adjuvant therapy for the prevention of impaired glucose metabolism in type 2 diabetes. Next is Professor Arun Lahiri Mazumdar. He is an INSA senior scientist, Division of Plant Biology, Bose Institute, Kolkata. He completed PhD in botany from Bose Institute, Calcutta University, and Doctor of Science in Biochemistry from Calcutta University in 1985. He has been shining in various posts like lecturer, reader, professor, and head of various universities in USA. And he is a fellow of National Academy of Science, India in 1996, Indian National Science Academy 2005, Indian Academy of Sciences again in 2005, West Bengal Academy of Science and Technology in 2011. His main research areas are plant physiology and biochemistry, abiotic stress responses in plants, plant biotechnology, and to name a few, he has a lot of awards and honors received in his career. He has received career award from UGC 1980 to 83 continuously. Sir Rashbihari Ghosh Traveling Fellowship, Calcutta University 1982, Overseas Associateship by DBT 1994, then Sadagara Swami Endowship Fellow Award in 94 to 95, then Rockefeller Biotechnology Career Fellowship, Parija Memorial Lecture Award from Utkal University, then Guha Research Conference. He was elected as Vice President for the Society of Plant Physiology and Biochemistry in 2001, then SB Saxena Medal Award from Indian National Science Academy 2008, S. Patnaik Memorial Lecture, Utkal University 2018. Our next speaker is Dr. Sanjay K. Banerjee. He's an associate professor from National Institute of Pharmaceutical Education and Research, Guwahati, Assam. He completed his PhD from AIIMS in 2004. His main work is mainly on molecular basics, and he has developed SGLT1. It is a novel glucose transporter in heart, statins as therapeutic targets for diabetic cardiomyopathy, and then role of vitamin D deficiency in cardiometabolic disorders, identification of one novel sulfur molecule or metabolites from garlic for heart failure, serum biomarker identification for diabetic patients at a high risk of CAD, TLR4 as novel target for cardiac fibrosis and heart failure. His main research interests are drug discovery, translational research in cardiometabolic disorders, especially focusing on cardiac hypertrophy, then heart failure, diabetic cardiomyopathy, NAFLD. And he has won a lot of awards: Fast Track Young Scientist, a CRB Award, American Heart Association Fellowship, Best Postdoc Award from University of Pittsburgh, Travel Scholarship from Keystone Conference, Postdoc Fellowship from Hillgrove Foundation, Ramalingaswamy Fellowship from DBT, then Professor Sailendra K. Vajpayee Award from Indian Society for Hypertension. He has more than 95 publications, 6 books chapters, then 14 review papers, and 8 patents.
Thank you. Thank you, Finos. Yeah.
Yeah. Okay, sir. Now I'd like to welcome Dr. Abhay Pandey to speak a few words, sir.
Yes.
Am I audible?
Yeah.
Yes, sir, definitely.
First of all, namaste to all my friends and young colleagues. So it is really a very good occasion to participate in the e-conference on current trends in life sciences organized by James College, affiliated to University of Calicut. I am thankful to Dr. Unni and his team, all of you, for inviting me to be part of it. And I'm really very happy to share all these functions along with you. So hopefully during few days, you will come across a number of lectures from distinguished scientists from different parts of India. And I'm sure students as well as scientists, they will get some inputs, good inputs for improvement of their, research prospects or any academic pursuits. So far, I know most of the participants are from UG, PG, and research area. So majority of the people, they are students, I must say. Few of us, maybe senior persons are there. So I would like most of our presenters, I'm just requesting on your behalf to most of the presenters to just enlighten the young students so that they can learn and they can easily take up the giveaway message. So I wish you all the best and thank you very much to all of you. Thanks to Dr. Finosi, thanks to Dr. Ponni, thanks to Dr. Naveen and all my colleagues sitting here. Thank you.
Thank you, Dr. Abhay Pandey, sir. Next, I'd like to welcome Dr. Prasenajit Manna to share a few words, sir.
Hello, am I audible?
Yes, sir, definitely.
Okay, thank you very much. I'd like to convey my regards to Dr. Unni and the Director Research of James College, as well as the Principal, Chairman, Vice Chairman, and also Dr. Finos, and also respected speakers. And it is, I'm very much privileged to be a part of this nice conference entitled Recent Trends in Life Sciences. And it is very much like the feeling proud to speak in front of the young generation, especially the students under the UG/PG classes, and especially like whenever I am talking in our earlier association with Dr. Uni in his earlier institution. It's very much like enthusiastic to answer their question from each small experiment and small queries, and that helps us a lot to learn more and particularly doing in some research area we are kind of more focused. But whenever you are going to speak in front of the young generation, especially the undergraduate students, so it gives you an opportunity to go back to your basics. And thank you very much the organizing team and for inviting me for this nice conference. And I'm looking forward for this speaker to learn their interesting topics. Thank you very much.
Thank you, Prasanjit, sir. Next, I'd like to invite Dr. Sanjay K. Banerjee for a few words. Sir, please unmute your phone. Sir, please unmute.
So now I'm audible, is it?
Yes, sir.
Okay. Dr. It is a great honor for me to be a part of this 10 days e-conference on current trend in life sciences. And I'm really feel that it will be one of the best conference. The kind of speaker you have chosen from different field, starting from plant biology, microbiology, and biotechnology, and disease biology. And I think we all can learn from the speakers and interaction with each other. And thanks to Dr. Unni for giving me the opportunity and inviting me. I was associated with Dr. Unni and in different way, one of the DBT project. I think we are 5 different institutes in Northeast. We are a part of the project, and we have not met, but we have a different interaction by phone and email. And I'm get the opportunity to touch with Professor Unni again. And thank you very much to become a part of this conference. Thank you.
Thank you, Sanjay, sir. I think our speakers are all right, sir.
You can go ahead with that because maybe somebody may be joining in between. So I think you can go
Oh.
ahead with the talk now. You,
Okay,
anything
okay.
else? You can go with the scientific session.
Yeah, Dr. Finos, you can also call Dr. Bulat Chowdhury for a few words.
Yeah, Bula Chaudhary, check the people who are there.
Okay.
Bula is here.
Okay. Okay.
Yeah.
Yeah.
Yes, yes.
Good evening, ma'am. Good evening, ma'am. Good evening, ma'am.
Namaskar.
Sir.
Actually, this is completely in a different purpose. So I just like to say, because these days, what is happening? We all are here in a virtual platform, right? And I think COVID is giving.
Ma'am, can you just unmute? Ma'am.
Yes. Okay. So actually, Namaskar. Good afternoon to all the respected dignitaries present in this platform and all the participants. So actually what is happening during this COVID period, lots of webinars are happening. So earlier what was happened, we were there inside a room, inside a hall, and we are organizing this type of webinar. So these things during this COVID period, I think it is good for us because we are sitting in the other part of the country and we were sitting there in the Kerala. But we could talk with each other and we could be in the part of this type of review. So thank you, James College, for introducing us in the other part of the country. And I have seen that the topic, the current trends in life sciences, is very relevant because this is related to UG, PG, or research scholars also the part of the event. And as I have seen, the organizing team has taken the speakers from almost all part of India and also from a different field of science. So that will be very much, that will be very good for all the participants in this event. And I expect the participants will be benefited from this program. And I wish the webinar will be very successful. And thank you for inviting me for this program. Special thanks goes to Unni sir because Unni sir always push us, always wanted to, always ask us to do something, do this, do that. And I met him in, I think in 2014. From 2014 onwards, he is always pushing us that you do that, you do this. So after going back to Kerala also in his own place, from there also he is also pushing us to do this, to come to this webinar. And so that's why I am very much thankful to Umi sir, and also as he has introduced me to the other part of India where I could meet you, I could talk with you, and further to collaborate with you. I could meet Dr. Finos, Dr. Mohan, Principal James College, Ali sir is there, Chairman James College, and all the professors and students of this college. And I would be happy to attend some of the lectures which is going to be here during this 10 days program. So thank you all, and I'm really happy to be part in this event. Thank you.
Thank you, Bula ma'am.
Finos, please check any speakers are there in the screen.
Yes, sir.
Before you go to the scientific session. Please check. Anybody is there? Did you find any?
No.
Yeah, Dr. Pranab Goswami is there.
Yeah,
Yeah,
Pranab Goswami. Bring him. Pranab.
Yeah, next I welcome Dr. Pranab Goswami to have a few words.
Yeah, yes.
Good afternoon to you all. Am I audible?
Yes, sir.
Okay.
Very
Good
much.
afternoon to you all. I am happy that I am invited to this e-conference, which is recent trends in life science research. I think it will be a very, very good conference, a very timely conference. It has been organized. And I am very happy that my senior colleague, Dr. B.G. Unni, who is a very good organizer, and I know him well for the 1986. And where Dr. Unni is there, I think there are no worry. Everything will be fine because he's a very good organizer. I know well. And so I'll be very happy to deliver this lecture. I am little bit actually engaged for the few days Otherwise, I could have given lecture today itself. But due to my pre-engagement, I could not come forward to deliver my lecture today or tomorrow. I'll be giving lecture. I think my slot is on 17th. So I think it will be great. And I congratulate all the first Dr. Uni, Dr. Finesh, and all these organizing members for organizing this excellent conference at the southern part of India. But although it is in southern part of India, it is encompassing all the eminent scientists and all these participants across India from in the length and breadth. So that is really good job you have done. So it is a wonderful, and I am looking forward to join with you on 17th of this month at 2:30 p.m. Right? Okay.
Yes.
Goodbye then. Thank you very much.
Okay, thank you, sir. Next,
Arulahari Majumdar was supposed to be there, but he said that he will be there. Is he there?
no, sir.
Okay. No. Okay. Check if anyone left.
Yeah. No, sir. I think we have. Yeah, we have checked, sir.
Yeah, go with the scientific session.
Okay, sir. Now I'd like to welcome Srimati Radhika R. Krishna, HOD of Department of Microbiology, for the vote of thanks.
Good afternoon to all. Respected Chairman Mr. Manalangureli, Vice Chairman Vasudevan Master, Academic Research Director Dr. B.G. Unni, Principal Dr. Naveen Mohan, Board Member Hamsa Master, and all eminent personalities and teachers and students from other institutions, and my dear students. It's my privilege to have a vote of thanks on this occasion. I extend a very hearty vote of thanks to the honorable delegates, Professor Abhay Kumar Pandey, Professor Dr. Girija, Dr. G. Mallikarjuna Swamy, Professor Pranab Goswami, Professor R.S. Verma, Dr. Devdatta Bhattacharya, Dr. Gulab Chaudhary, Dr. Prasanjit Manna, Professor Arulahari Majumdar, Dr. Sanjay K. Banerjee, who blessed us with their presence and took out valuable time of your busy schedule. Thank you. I must mention our deep sense of appreciation for our academic director, Dr. B.G. Unni, for organizing such an amazing program. Thank you. Also, I would like to extend a special thanks to Mr. Chairman Manalangureli, Vice Chairman Vasudevan Master, Board Member Hamsa Master for inspiring and giving us an opportunity to organize these.
Some problem with the link, looks like.
Okay, sir. Okay.
Yeah, we will move on with the next session.
Okay.
Dr. Finesh.
Thank you, Srimati Radhika. Next we can move on to our next session, which is the inaugural session and scientific session, which will be dealt with Abhay Kumar Pandey. So I would like to welcome Ms. Shinsi P, who is the HOD Department of Biotechnology, to welcome the speaker.
Good afternoon to all. It's an honor for me to introduce today's guest speaker, Dr. Abhay Kumar Pandey, Professor of Biochemistry and former Head of Department of Biochemistry, University of Allahabad. Dr. Abhay Kumar Pandey's career has taken through more than 25 years of teaching and research experience. He holds a number of positions like Fellow of SPR, member of many academic bodies, including the National Academy of Sciences, Society of Biological Chemists, Indian Science Congress Association, and Italo-Latin American Society of Ethnomedicine. His research activities are focused around oxidative stress and free radical biology, biochemical toxicology, antimicrobials, and cancer biology. He has more than 90 publications to his credit, including many research articles in international journals of repute, and chapters in the books published by Springer, Elsevier, and other publishers. Based on Google Scholar citations, Professor Pandey has more than 4,600 citations. One of his review articles, Chemistry and Biological Activities of Flavonoids, an overview, has been cited more than 2,400 times. Today, all of us are elated to hear his lecture. Now, I would like to welcome Dr. Abhay Kumar Pandey for his presentation. Thank you.
Thank you, Dr. Professor and Head of the Biotechnology Department. So at the outset, I would like to thank Dr. B.G. Unni. Especially, I would like to mention about him that he has been alumni of our institution during early 70s. So he, from Kerala, he traveled to Allahabad and he became our integral part, just like nucleus of a cell. So we love him a lot, and we always get reciprocated from his side. So I'm so nice to be part of his company. And after retirement, I have just seen that he has joined James College. So I am sure under his able guidance and leadership, what he has just conveyed with him will definitely provide some newer insight for the development of James College under Calicut University. He is man of principles and he is man of technology. So looking at his track record, I am sure your college will get some further heights in due course. So I am thankful to you, sir, and I just wish you continue giving some good thoughts to the nation as well. So today's talk, I think at the same time, I am thankful to Dr. Finose. He has been continuously in touch. Yesterday he rang me, day before yesterday he rang me, and today also I got his message. So I am thankful to Dr. Finose for continuously just taking care of this program. And moreover, I would like to thank especially the chairman, Mr. Ayub, who has taken pains to build this GEMS institution. He has contributed a lot, and I extend my thanks to Vice Chairman, your principal, Dr. Mohan, and all teacher fraternity. Now, I think I must begin my talk. So I am just opening the PowerPoint presentation. Hope I am audible.
Yes, sir.
Yes, sir. Yes, sir.
So I'm now sharing my slides, and topic of the talk is oxidative stress and associated complication. So I think the topic is quite relevant in the time of
the slide has not yet come. The slide has not yet come.
Slide is not visible, sir.
No, not visible. Not visible. You have to share.
Sir,
below I think there is a button to share the slide. Then only it will come.
Oh, okay. Yes, sir. Just I have just a minute, sir.
Yeah.
For share, I have to click more options, I think. Settings, report. This, can you help me? How
There
can
will be,
Sir, Abhay sir, there will be a button called present now in that, in the right corner.
no, I have clicked it.
Okay.
Yes, then change layout, full screen, turn on caption, settings, use phone, report the problem.
Yeah.
Report
You can do full screen, sir. Go for full screen, sir.
Okay, okay. So now this is share. Just a moment. I am just taking help of my
Sure, sir.
slide. Just help me.
If you have a problem, he has got his slides. You can share from there.
Yes, sir.
Okay, I'll do it.
Yeah,
Yes,
that you can do it from there.
Yes. Share. Let's go on. Maybe some technical glitches there.
Yes, sir. Please wait, sir. I'll do it from here.
Okay.
You can just go on with the presentation. I'll just change the slide, sir. Please wait.
Okay.
Slides will be shown here, so there is no problem. You will get it.
Yes, sir. Okay. Visible?
Yeah, it's coming. It will come.
Yes. Now it is visible. So, the topic of my talk is oxidative stress and associated complications. So, at the outset, I think it will be quite pertinent to talk about stress, especially during the corona times. People who come across the corona patient, they are highly stressed. If they come to know that they have developed corona positive, so you can feel what type of stress is there inside their body. So obviously, whenever there is stress inside our bodies, certain metabolic changes at biochemical level, at gene level are taking place. And they are transcribed and translated in the form of some specific symptoms, which give us certain problems. Now, I'm talking about this oxidative stress. It is an imbalance between systemic manifestation of reactive oxygen species and the biological system's ability to readily detoxify the reactive intermediates or to repair the resulting damage. Means when we are talking about reactive oxygen species, it is not simply ROS, we also include reactive nitrogen species. So when we talk about ROS or RNS, they fall under category of free radicals. So we know a lot of free radicals are produced inside the body, and our body has mechanism to counteract the excess of free radicals. Otherwise, if we go through biological science, we find that these free radicals also act like signal molecules at lower level. So their requirement is very much visible in our body, but at higher concentration, they are responsible for creating havoc. So we have to get rid of that. Then, if we talk about the free radical, so free radicals, I think everyone is knowing that any atom, molecule, or ion containing one or more unpaired electrons. So, as we know, the free radicals are very reactive and highly unstable. They want to get stabilized by just taking electrons from some other sources. So if we talk about their properties, they are highly reactive. At the same time, their half-life is very low, very short half-life. And if they attack, just like nuclear reaction, they also start a type of chain reaction, and that chain reaction is responsible for propagating and generating new radicals continuously. And these radicals are responsible for causing damage to biomolecules, cells, and tissues. Now, when we talk about reactive oxygen species, so what is the source of ROS or RMS inside the body? In this slide, you will observe A, B, C, D. 4 parts are there. So we know that we are continuously taking in oxygen. So 4 electrons are required for reduction of oxygen. And more often we come across when oxygen accepts electron, it is converted to superoxide radical. Means it is O2 having lone pair of electrons, negatively charged. So it behaves like radical. As we have seen that radicals are quite reactive. So it might react very actively. But at the same time, these superoxide radicals, they also go and associate with hydrogen atoms, and they are converted to hydrogen peroxide. Again, as though hydrogen peroxide is not a radical, but still it behaves like a free radical. Hydrogen peroxide has got property of producing a highly reactive radical, that is hydroxyl radical. It breaks down into 2 hydroxyl radical. At the same time, this hydroxyl radical may be converted to water. Means this is one part. Then when we talk about the reactions of H2, here I would like to mention that hydroxy radicals are very dangerous so far their activity is concerned. Their half-life is very small, so they cannot create much havoc. Had they got more time, they would have created really disaster. So their another product is H2O2. We know hydrogen peroxide is product of dismutation. Means superoxide is dismutated, especially in living system, by superoxide dismutase. It is converted to this free radical is converted to H2O2 and oxygen. Then H2O2 is again through homolytic fission, it is broken down to hydroxyl radical, and through other this reaction you also find number of radicals. Then third process, we know transition elements, iron, manganese, copper, number of these metal ions are present throughout our body, and they are more often part of our enzymatic machinery. So they have one specific attribute. They can either take or give 1 electron anytime. So this ability of giving or taking electrons makes number of you can say molecules vulnerable to attack by free radicals. Because if iron reacts with the hydrogen peroxide, again we get production of hydroxyl radicals. So in this way, Fe2 or Fe3 they go on interacting, and they continuously produce number of radical atoms. At the same time, if we talk about the attack by hydroxyl radicals, say for example lipid membrane in the T, at the bottom section one can see the RH plus hydroxyl radical. RH is nothing but any organic material having hydrogen attached. So if we assume it to be lipoid membrane, so in lipoid membrane we get CH2 type of methylene bridges. So if it reacts with hydroxyl radical, we get again generation of some lipid radical, organic radicals, they are produced. And we get continuous propagation of radical chain by producing peroxyl radical, RRO, then again organic peroxides and RO alkoxyl radical. So these are the processes through which variety of free radicals, or what we call reactive oxygen species or reactive nitrogen species, are produced in the system. Now, free radicals' life is very crucial for their good or bad action. Now, in this slide, we can observe that hydroxy radicals have half-life of almost, longevity of nanoseconds. And rest of the radicals are in microsecond or seconds. So hydroxy radicals, they are produced inside the mitochondria. So within nanosecond, they can cause damage. Means wherever they are produced, if some biomolecules are there in its vicinity, they can damage that thing. But other radicals, they take some time. Here, if we talk about hydrogen peroxide, its longevity is for minutes, though it is not very active. But still, when we talk about the minutes, it can pass from cytoplasm to nucleus, and even inside the nucleus, it may create DNA damage. So hardly, longevity hardly matters. Sometimes even less reactive metal can also create more problem. So most of the free radicals, being superoxide, hydrogen peroxide, singlet oxygen, or lipid peroxide, they all are responsible for creating damage if they are present at a certain critical level. In the bottom part, it is given that role of transition metals. As I have talked earlier, the transition metals are very active in taking or giving electrons. So if they get any starting material, they can convert it to some more reactive species. So we get further action of those radicals. Now, at this point, it is pertinent to know what are the effective endogenous sources of ROS and what are exogenous sources. So when we talk about endogenous sources, we know through electron transport chain, electrons are passed from different complexes up to the production of ATP level. Now what happens? It is assumed that all electrons, while passing through this this electron transport chain, they will produce ATP, but up to 2% electrons, they somehow they prematurely and incompletely they are reduced to give superoxide radicals. Means approximately 90% of oxygen is consumed for ATP production, and only minor fraction, less than 2%, is responsible for production of radicals. Second part is when we talk about the steroidogenic system, especially ovaries and testes. There we also find the electron transfer reactions are catalyzed by mitochondrial P450 system, and they take help of NADPH. And during this process, some electrons leak, they are leaking and they are reacting with oxygen to produce superoxide radicals. Now, if we think about this ovary and testis, because the germ cells are present, if they are exposed to the radical, so a lot of damage will be there. So to counteract that damage, ovaries and testes are having a lot more antioxidants of natural origin, be it vitamin C or other antioxidants in them. So these antioxidant systems, they are responsible for counteracting the excess superoxide or hydrogen peroxide produced inside the steroidogenic tissues. Then ROS or free radicals are produced in immune cell signaling via NOx pathway. So if we talk about neutrophil action, eosinophils, mononuclear phagocytes, we also find the free radicals are produced upon stimulation. Here I must say that production of free radical is essential up to a certain extent. It is, at the same time, it is also necessary. But if somehow it goes beyond the level, then it becomes dangerous. So when we talk about exogenous sources, different type of pollutants, heavy metals, tobacco smoking, drugs, smoke, xenobiotics, and ionizing radiations, they are again responsible for production of radicals. If we talk about ionizing radiations, what happens? It is the radiolytic process that is responsible for production of free radicals. At the same time, xenobiotics are metabolized through our liver system, and the liver, they produce free radicals. In the same way, every external xenobiotic material is treated by our liver, and at one point or other, we come across certain radical sources. Now, these are some pictographic representation of some cellular sources of ROS and living cells. So what is mitochondria? We know from ubiquinone, first and third complex of respiratory chain, we find superoxide and hydrogen peroxide is produced. Then in phagocyte itself, we can see the NADPH oxidase system is there, that is also responsible for production of the radical. At the same time, if we talk about the membranes, 5 lipoxygenases, they are also responsible for production of free radicals. So if they are produced in a balanced quantity, they act as signal, but if they go beyond, they prove fatal. So this is again a little bit repetition of the earlier slide, the generation of free radical through non-biological and biological pathways. We have already talked about the sources, external sources, internal sources in inflammatory cells, in fibroblasts, endothelial cells, respiratory chain, xanthine and NADPH oxidase. So superoxide radical is produced, it is converted to hydrogen peroxide, and through our antioxidant enzyme system, which are present in our cells, that is catalase, glutathione peroxidase, glutathione reductase, H2O2 is converted back to water molecule. And there are a number of SODs, copper zinc SODs, manganese zinc SODs, extracellular SODs, they are present. At the same time, NO, nitric oxide is also there. So when it interacts with superoxide radical, ONO type of radicals are generated. And through Penton reaction, they can produce hydroxyl radical, which are ultimately responsible for cell destruction. Now, if we look at this slide, ROS has been produced through different sources. Now, what are the targets? Here we see nucleic acid, proteins, lipids, and carbohydrates. Means all the biomolecules which are present in our system, they are targeted in one way or other way by these free radicals or reactive oxygen species. When we talk about the nucleic acid, there is a possibility that it could be acted on epigenetic machinery. We know in epigenetic machinery, DNA methylation is there. So if we talk about 5-methyl cytosine, that is present. Sometimes, just because of the free radical, this methyl group, cytosine is converted to some other pyrimidine compound, and that ultimately is responsible for creating a pyrimidinic site. So that type of process is responsible for production of what we call mutations. So in this way, it is responsible for damage. At the same time, if methylation or demethylation is going on, means somehow we are interacting with our cellular machinery regarding expression or just subsiding some processes of expression process. So all these damages at nucleic acid level, maybe protein, when we talk about protein, amino acids undergo oxidation, lipids undergo oxidation, lipid chains are broken down, carbohydrates undergo oxidation, advanced glycation products are produced. We see that advanced glycation products, they just club with proteins, and especially in diabetic individuals, they are creating a lot of problems. So all these activities of free radicals, they are responsible for converting our biomolecules to a different form, which is responsible for ultimately the damage. And this damage results into impaired physiological function. It might lead to age-related diseases. It might progress aging, or at the same time, it might lead to a cell death. So everything is possible. So this is free radical production in mitochondria. SOD is there, H2O2, then Fenton reaction, just like iron, catalysis, everything is there. So in mitochondria, we can also get the protein, lipid, DNA, oxidative damage. Now we know that peroxisomes are sources of ROS and RNS. So number of enzymes are present in peroxisomes. And if we go to the bottom left, we find during fatty acid metabolism, acyl coenzyme oxidases, they are responsible for production of H2O2. At the same time, D-amino acid oxidases or aspartate oxidase, urate oxidase, xanthine oxidase, NO synthase, all these enzymes, they are producing mostly H2O2, some are producing superoxide radical or nitric oxide radicals. At the top left flank, we can see number of oxidases are present in the cell, and these oxidases, especially when we talk about NOS, nitric oxide synthesis, they are responsible for production of nitric oxide free radical NO radicals from conversion of RGB. And these nitric oxide free radicals further produce a different type of radicals. So we get continuous influx of radicals in our system. Now, this is, I'm going to show that during mitochondrial electrons transport chain, how electrons are transported, how ATP is synthesized, and in the due course, we find production of free radical. And if these free radicals get time, they are responsible for damaging our complexes which are present nearby. Yes, you see, Krebs cycles is running on, NADH is being oxidized, electrons are being passing through Q, different 1, 2, 3, 4, 5 complexes we can observe, and ultimately oxygen is taken, it is converted to H2O2, and after passing through the fifth complex, ATP is synthesized. So during this process, electron has been transmitted from complex 1 to complex 5. Now you see, from first, third, fourth, everywhere, number of entities are coming and they are responsible for production of superoxide radical inside mitochondria. Now, what happens when superoxide radical is produced in the mitochondria? There is GPS, glutathione peroxidase. There is catalysis, means there are two pathways for conversion of H2O2 to H2O. One is through catalysis. Catalysis is directly responsible for breaking down H2O2 to H2O plus oxygen. But GPS takes help of glutathione. We know a tripeptide, very important antioxidant tripeptide present in the system, that is converted to GSSG, means oxidized glutathione, and that oxidized glutathione is reduced to, again, reduced glutathione by glutathione reductase in our system. So here we see this hydrogen peroxide in presence of, again, I'm going back, this thing, hydrogen peroxide is mixing with the iron and it is responsible for production of hydroxyl radical, and this hydroxyl radical goes and attacks the complex, especially in the mitochondrial membrane. So in this case, if we are getting larger production of hydroxyl radicals, they are responsible for breaking down our lipoid or protein, which are whatever is present in our membrane, mitochondrial membrane, and ultimately cell death is there. The next, free radicals and mitochondria, again, another animation is there. If we see this from superoxide radical, we are generating H2O2, and from H2O2, with the interaction of iron, we are getting hydroxyl radicals. So this hydroxyl radical, they might attack at mitochondrial DNA. At the bottom part, you can see. Then again, iron sulfur centers are there in equinitase enzyme. They might attack. Because of this hydroxyl radical, oxidation of proteins take place. Once the amino acids in the proteins, they get oxidized, the activity of enzyme declines. It changes. So we find a different scene if we are getting this type of attack by free radicals. Just see, this is the cycle of free radical attack on mitochondrial DNA, on Krebs cycle on cell membrane. And what happens? This keeps on going. Ultimately, electrons are excluded from MPTP, ANT, VDAC. It comes into the cytoplasm, and finally, this is responsible for activation of caspases. Inactive caspase is made active, and further it leads to apoptosis. So, free radicals have different tasks under normal circumstances as well as under abnormal circumstances. Now the question in general people talk about, generally people think that free radicals are enemies. So in my opinion, or as a biologist, nobody will agree with this opinion. Because here the front line is free radicals, frames are foes. So we know free radicals act in both the fashion. They can behave like friend, they can behave like enemy. So first, I'm talking about the beneficial roles of free radicals. We have already seen in the last animation that during the generation of ATP from ADP, through oxidative phosphorylation, free radicals are generated. But if you see, through generation of ATP, we are producing a small amount of free radicals, and that amount is essential. So under controlled condition, this is beneficial aspect because if we stop the pathway of electrons, we can stop the production of ATP. We will not get ATP produced at all. So obviously, to get ATP, we have to produce some free radicals. But at the same time, our machinery, living system has got system to counteract the overload of free radicals. So in generation of ATP, we find help. Then detoxification of xenobiotics. Any foreign compound which is taken by us in our system, they are detoxified through cytochrome P450 system, and there again free radicals are produced, and their role is very important in this. In the last animation, we have seen apoptosis, especially free radical generation, is accountable for apoptosis of defective cells. Then one very important question is the killing of microorganisms, especially during the infection, and cancer cells by macrophages and cytotoxic lymphocytes. So during that process, again, a lot of free radicals are produced, which are responsible for killing the microbial cells present in our living system. At the same time, oxygenases, cyclooxygenases or lipoxygenases, they are responsible for special signal molecules, which we call just like hormone, they are prostaglandins and leukotrienes. So they are very important regulatory compounds. So during their production, we also find free radicals are produced. So we cannot escape production of free radicals. In other way, these are the beneficial aspects of free radicals. Then some additional beneficial roles. I am highlighting the presence of low concentration of free radicals is important. Time and again, I have emphasized on this aspect. But lesser concentration, lesser load of free radical is necessary for normal redox states, immune system functioning, and intracellular signaling in the body. But at the same time, if we talk about the immune system, neutrophils and macrophages use reactive oxygen species to destroy engulfed microorganisms. In the last slide, I have discussed. So there, what we get superoxide radical and hypochlorous acid, HOCl, a very strong oxidizing agent, is generated in the macrophages that is responsible for ultimate degradation of microbes. So here one can see the reaction, how it takes place. From oxygen, we are producing superoxide radical through eradicate oxidase reaction. This is converted to H2O2 by SOD, and then H2O2 can be transformed to HOCl by myeloperoxidase reaction. So myeloperoxidase reaction is a very essential reaction for getting rid of microbial cells from the living system, because by this we can remove the infection cycle. So here, these are important beneficial attributes of the free radicals at certain level. Then these are negative attributes. In negative attributes, we find the tissue damage by radicals. As we've already discussed, that if DNA comes into contact with the free radicals, so obviously free radical oxidize. It may oxidize DNA bases. It may oxidize what we call ribose. So just because of this oxidation, there is a possibility of single strand breaks. And further, it might be responsible for double strand break as well. Then if single strand break takes place, again we know the repair reaction, repair mechanisms are there. If there is some defect in repair mechanism, that might cause heritable mutations in germline, especially in germline cells. And in somatic cell, we can find cancerous growth. At the same time, we know that lipids are very strong, attractive target of free radicals because of their polyunsaturated fatty acid contents. So they undergo oxidation and they destroy lipid membrane. Lipid peroxides are also involved in atherogenesis. Means again, if we talk about the association of this oxidative stress, so we come to cardiovascular diseases. Means it is manifested in the form of atherogenesis. Maybe some blockages are generated by suppose LDL is there, LDL is oxidized, so oxidized products of LDL, they are responsible for formation of plaques at the endothelial layer of our blood vessels. So that might be responsible for breakdown of our cardiovascular system. At the same time, we talk about the endothelial system. We know in endothelial system, nitric oxide is produced, NO, and that is responsible for vasodilation. But we know when free radicals attack, there is an adverse reaction. So during reperfusion process in atherogenesis, there is again further possibility of free radical production. So atherogenesis is a common result leading to myocardial infarctions and other problems. And lipid peroxides break down to bialdehydes with 5 protein. And another important attribute of the free radical damage is that oxidation of amino acids. We have also discussed in previous slide in a different respect, but here we are talking in a different reference, that oxidation of amino acid protein may lead to formation of antibodies against modified proteins. Means once the protein is oxidized, that is not recognized by our own system as self-protein. So we might develop autoimmune type of factor. So we may produce antibodies and ultimately autoimmune disease results. At the same time, oxidized amino acids may catalyze further formation of oxygen radicals. And in the last line it is written, oxidized amino acids in LDL are involved in atherogenesis. So obviously, atherogenesis through lipid peroxidation we can get, through oxidation of amino acids present in LDL, they all contribute towards the cardiac anomalies. So if we look at a general slide, so free radicals and diseases. So generally, as we know, free radicals are highly reactive. They attack fatty acids, DNA, cholesterol, or any biomolecule present in the cell, and a rapid destruction of destructive chain starts. One free radical attacks some biomolecule, then another radical is produced, and this chain keeps on going. And ultimately, if this chain is not stopped, what will happen? This will result in injury to tissue. So what will happen? This will damage membrane lipids and proteins. So number of protein enzymes or membrane fractions, they will be damaged. Then we might get precancerous changes, means beginning of cancer. At the same time, cholesterol oxidation may start cardiovascular problem. So ultimately, end result of this continuous chain reaction is the development of cancer or heart disease or macular degeneration or other diseases or aging. We talk about the aging, we find continuous accumulation of free radical and their products that is responsible for aging of our cells, our whole body system. So on the lower slide, we observe that in vitamin C, it is shown as a wall between different plates of membranes. So if free radical attacks, number of our lipid molecules are broken down, but when vitamin E comes, it again takes electrons, gives and takes electrons accordingly, and it stops the reaction, free radical. One very important aspect I would like to highlight about this, if we simply give vitamin E, after some time vitamin E is converted to a free, radical, that is a stable radical and that is not harmful. But for regeneration of vitamin E, we always need vitamin C. So in our body system, living cells, vitamin E, vitamin C cycle continuously goes on. Vitamin C is responsible for regeneration of the native form of vitamin E, which is again stopping this reaction. So in this slide, free radical toxicity, whatever we have, most of the things we have talked about, it has been shown here. Means from xenobiotics, number of reactive oxygen species are produced. Then oxidated nucleic acids, we find one very important aspect that 8-hydroxyguanosine is produced. So if 8-hydroxyguanosine is produced in our nucleus, that might lead to mutation. At the same time, we find production of isoprostane. You know, arachidonic acid, if we talk about the arachidonic acid, integral part of the membrane, more than 20 carbon lipid material. So from arachidonic acid, we find isoprostane synthesis in presence of reactive oxygen. So this isoprostane is nothing but free radical toxicity product, and we can identify. So in this slide, simply free radical toxicity has been shown. Here again, almost similar thing at more molecular level is shown. That is 5-methyl cytosine undergoes oxidation on left flank. You will find it is converted to OH and O group through successive oxidation. And on the right flank, we find oxidative deamination is responsible for conversion of 5-methyl cytosine to thymine. So again, our repair system is responsible for accommodating some repairs, but if they are not continuously accommodated, we might undergo some mutation process. This is complete summary of what we call free radical toxicity. I think it is not required because we have already talked in greater detail. But in this slide, all processes have been put at single place. Now, when we talk about free radicals, they are also responsible for altering our gene expression. In this slide, we can see superoxide is converted to hydrogen peroxide. It gets into the cell. Hydrogen peroxide, they may activate different type of enzyme systems, and by activating enzyme system, it can bring about the genetic expression alterations. At the same time, it can activate different proteins such as guanyl cyclase, ion channels, or phospholipase. So in this way, by changing redox status, we get change in altered enzyme activity. At the same time, we get change in genetic expression pattern. And that might culminate in different type of pathological symptoms, what we are observing maybe in cardiovascular disease, in diabetes, in cancer, or many other diseases. So, what are the things which counteract this free radical? So, antioxidants, they are the substances used for neutralizing free radical and their actions. In humans, we have evolved a highly sophisticated, complex antioxidant protection system. Now, this complex system consists of small molecules such as glutathione, vitamin C, vitamin E. Then we talk about some enzymes, SOD, catalase, glutathione peroxidase, glutathione reductase, etc. So that is responsible for protecting the living system from free radical attack. So it means that antioxidants are capable of stabilizing or deactivating free radicals before they attack. Means antioxidants can play their role at the beginning. They can stop the reaction beginning, or they can intercept the reaction when it has started, or thirdly, if the product has been formed, it can still deactivate further. So antioxidants are absolutely critical for maintaining optimal cellular and systemic health. In this slide, we find different type of enzymatic and non-enzymatic oxidants are shown. So primary enzymes, what we have discussed, SOD, catalase, glutathione peroxidase. Secondary enzyme, glutathione reductase, glucose 6-phosphate dehydrogenase. So just we know that glucose 6-phosphate dehydrogenase is present in a cycle that is responsible for production of NADPH. So we know what is the significance of NADPH, because in cell metabolism, reducing equivalents are always provided in synthetic pathway by NADPH. So NADPH is very much necessary when we want to provide hydrogen to GSSG, means oxidized glutathione. So from NADPH it comes, and NADPH cycle should go on. So these are secondary enzymes. Then non-enzymatics, we talk about minerals, vitamins, carotenoid, low molecular weight, and certain other factors. But in addition to non-enzymatic antioxidants, we also talk about natural products, especially the polyphenols. So if we talk about the polyphenols, we come across flavones, flavonols, anthocyanidins, number of compounds, phenolic acids, etc. So nowadays, if we are talking about the green tea, we get a specific epigallocatechin gallate in that green tea that is responsible for providing us the antioxidant action and keeping ourselves fit. At the same time, quercetin, camphorol, hesperidin, number of polyphenolic materials, they are providing very good antioxidant material. How do flavonoids or polyphenols act as antioxidant? What makes them a material for antioxidant? Now, if you go through the structure of flavonoids, you will find, or in general, not only flavonoid, polyphenols, we have number of hydroxyl groups. So whenever these compounds come in contact with the free radicals, they donate their hydrogen. Free radicals receive hydrogen from them, and in turn, intermediate radical type of structure is generated for flavonoid or other phenolic compounds. And then again, in the second reaction, again it interacts with the free radicals. It also provides additional amount of hydrogen. So R is converted to RH, and here a quinone OO, we can see this structure in this slide, of stable structure is generated. So the disastrous action of free radicals is set by providing hydrogen atom, and in turn these become stable, and at the same time they provide protection to our biomolecules by getting themselves oxidized. By getting themselves oxidized, it means they are donating hydrogen atom. At the same time, we know just through the coordinating attribute towards the metal ions, they can chelate metal ions. If excessive metal ions, as we have talked about, the transition metal ions, they are excessive, they present in excessive amount in our system. They are also responsible for production of free radicals. So these polyphenolic compounds, they chelate these metal ions, and thereby our system is protected. Now, if you talk about this metal toxicity, one very common thing we have observed, if a pregnant lady goes to a doctor, always she is prescribed to take folic acid, vitamins, and some iron tablets. In earlier days, when people were living in villages, I don't think people were just taking more amount of iron. So this type of excessive iron might create problem in some cases, because if free iron is present, it is not associated with any protein or other moiety that is free to react. So there is a possibility that it will create free radical, and especially in the womb to the newborn, it might create some problem. It might alter the genetic constitution, creating some mutation. So we find overload of any metal is also equally disastrous. So we must avoid overload of metal ions in our food as well. Here to counteract this oxidation reaction, we have talked about flavonoids. They are natural products. So question is, why do we rely on natural products? This is important aspects. First thing, that nature is endowed with vast majority of plant species, and they have apparently numerous compounds. We cannot even count those. So In other words, we find lot of structural diversity in nature. So they provide us diverse compounds. Apparently, unlimited quantity is also there. So that is available to mankind. Now, whenever we are talking about the third point is the evolution against challenges. Even if we talk about the evolution of human being or any cell, we know during the evolution process, our cells have struggled, and whatever good has been kept inside the cell, that is responsible for increasing our longevity. So these natural products have evolved against several challenges. So they have inherited some very good chemical components. They are providing advantage to fight other conditions. At the same time, they are quite potent. Generally, they are linked with the safety and efficacy as well. And what are sources? We know plant, microbial sources, animal or marine one. It can provide us. Now, we know most of the Indian population, not only Indian population, but other third world countries, they are mostly dependent on alternative system of medicine, be it Chinese medicine, be it Indian system of medicine, Ayurveda, be it homeopathy. I mean, different type of traditional systems are there. Now, if we look to the world herbal market, as for 2009, there was $154 billion trade, and which is anticipated to increase to $212 billion at approximately a rate of approximately 4.5 to 5%. Increment is there, and this is accountable for herbal supplements which are being sold throughout the world. This is $1 billion, means $150 billion to $200 billion rise is there anticipated. In this context, if we compare the Indian Ayurveda market, it has traded worth INR 300 billion. These 300 billion rupees of trade was there in 2018, and from 2019 to 2024, there is anticipated increase of almost 400 billion Indian rupees. So there is again continuous increase. And as per our government's guidelines, we want to be self-sufficient in number of processes. So looking at the what we call natural wealth in India, we might find it could be a better source if we are going to trade throughout the world and providing our natural products. If we look at the Indian plant market, there are a number of industries, only few of them I have just written here. Dabur, Patanjali, Himalaya drugs, and all this. This is simply the names. In addition, you can find many more hundreds of companies are there which are selling their products inside India as well as abroad, and they are getting revenue. While talking about the plant products, we always talk about synergy, because most of the effective phytomedicines in the market are as whole extract. If we look back to Patanjali product or Dabur product or Himalaya drug product, even we talk about LIV52, we know a very good drug for liver. That is simply a conglomerate of number of plant extracts put together. So any compound, even if I am giving an example of this LIV52, it is prescribed by all allopathic doctors along with other drugs. So that is the beauty of our Indian system of medicine, you can say. So when we talk about the synergy, we can find it in these type of preparations. So synergistic action might be responsible for sometimes better result as compared to a particular part. So there are a number of evidences to this effect as well. So I would like to just show a little bit of my research. I'm not going in detail because I wanted to share most of the basics of free radical biology to the student community. So here, just before going to few research aspects, if we are taking medicinal plants, in the topmost slide, we find different type of species are shown. So mostly trees and herbs are preferred as compared to shrubs and climbers. This is based on some data. At the same time, in the bottom side, right side, you will see that the fruits, leaves, and seeds are accountable for most of the ethnomedicines as compared to underground or other parts. So what my logic is that fruits could be easily plucked, leaves could be easily plucked, seeds can be easily harvested. So that is why most accessible parts are utilized for production of large number of medications. So in some of our tests, especially in our laboratory, we are going for natural products, in vitro, in vivo, both type of tests we are performing, and mostly on biochemical toxicology associated. So we try to see if the plant is having free radical scavenging action or not. In other words, it is providing protection against free radicals. So we have observed the number of plant extracts. If we have sequentially extracted extracts in different solvents from non-polar to polar, we have found diverse extracts. They show quite similar activity. What do we interpret from it? Means when we are extracting anything in different solvents sequentially, there is a possibility that like dissolves like. Means in the non-polar material will go in non-polar solvent and polar material will go in polar solvent, and in between we find some transient population of non-polar and polar exists in between. So during this segregation, different molecular species are segregated. So sometimes we find some specific extracts, such as in Solanum xanthocarpum, we find most of the extracts have shown very good DPPH free radical scavenging activity. Same thing lies with the T. cardifolia. So all these activities, they are showing a good potential for scavenging action against the free radical damage. In vitro, I'm not talking about here in vitro. At the same time, reducing power is also a method to see how easily a compound can donate hydrogen. Means loss of hydrogen, what is that? Loss of hydrogen is oxidation, gain of hydrogen is reduction. So reducing power says that plant extracts have ability to provide reducing equivalents to the free radical, and by getting that thing, reducing electrons or hydrogen, they are stabilized and they further do not go for any reaction. So in most of the plant extracts, E. cardifolia, Parthenium or Solanum xantho, are a number of extracts. Here we can see in some of them aqueous extracts are doing well, in some of them acetone extracts are doing well. So these are depending on the characteristics of the compounds present in these extracts. If you remember, in few, I think in one of the slides, I have shown the mechanism of action of flavonoids. So metal ion chelation was shown there. So by chelating metal ions, we can stop further free radical generation. So we have also tested metal ion chelating ability of different compounds, different extracts, and sometimes we also check it for pure compounds as well. So accordingly, we find some specific extracts in T. cardifolia or Solanum xanthocarpum or Parthenium. They are responsible for showing greater and greater metal ion chelating activity, indirectly showing their radical quenching action. So this is also beta-carotene bleaching linoleate model. In the next slides, few slides I am going to talk about, we also feed some genobiotics to living system, especially rat, and we want to see the efficacy of some natural products, whether it is rectifying the error, harm done to the living system or not. So these are few free radical associated liver toxins, ethanol consumption, chemicals, CCl4, aflatoxin, anything, or any drug. What we are giving, that is obviously has to be metabolized by liver. So obviously liver is at the first level of you can say activity of damage. Then some infections. So all these processes are responsible for liver damage. And in these processes, is there a possibility of reducing these damaging chances? So here, if we are giving these drugs, they are producing reactive metabolites. Especially, there are a number of drugs which, when they are taken in the body, in the liver, they are some of them they are metabolized to more active products, and some of them they are metabolized to less active. So depending on the type of drug, we find some reactive metabolites. They can go and make protein adducts. They can be responsible for depletion of our glutathione or the tripeptide, and ultimately they might lead to oxidative stress, and this might proceed to apoptosis or necrosis. Necrosis means more rapid damage to cells as compared to apoptosis. Apoptosis is a controlled damage. So in the same way, after this, again we find a lot of damage to copper cells. So this is simply the liver detoxification pathways. So phase 1, phase 2 enzymes are there. We find at number of stages free radicals are generated, and they are responsible for damaging our system. So this is biotransformation. How does it take place? So during our experiments on isoniazid, we know that it is taken by the patients who are suffering from tuberculosis. So they take isoniazid, pyrazinamide, etc., ethmobutyl for very long term. So we have observed that some of the plant extracts, they provide certain level of protections to liver if we are taken together with the isoniazid for long term. So here the biochemical parameters, creatine, bilirubin, ALP, GOT, GPT, hemoglobin are shown. So here if we talk about the control and INH, so as compared to control, means INH means isoniazid fed rats, one can find the altered levels. But when we further one control, we have taken LIF52, then other plant specimens. So we have seen that some of the plant specimens, especially at the right plant, Iconia, it has provided some better degree of protection, while the one other plant, Syngonium, could not provide any protection at all. At the same time, we have also seen the Piper longum, most commonly used in household, Piper longum. So it is also responsible for providing hepatoprotective effect, and it could be monitored with the help of biochemical indices. Same thing with cinnamaldehyde, again a natural product present in cinnamon. We have also studied the methynyl aloe. You know, methynyl aloe is a colored material, organic material that is yellow in color. Sometimes it is mixed with food material, maybe in laddu or some other yellow products. So if it goes in larger quantity, it might create liver anomalies. So in that case, we have also found, especially the Indian foods, we know a lot of spices are there, so number of natural compounds are present in there. So we automatically get protection out of it. So in this slide, we are showing the effect of cinnamaldehyde, the natural ingredient present in cinnamon and other things. Here again, same story. So cinnamaldehyde has shown quite good results in methynyl yellow fed rats. If when we talk about the histological section, we have observed quite good improvement. We have also observed some effect on the cancers, especially on cell lines. So these are the hallmarks of cancer. I am just scrolling rapidly because I think I have taken a lot more time. So plants have number of constituents. They might act at different level for inhibition of cancer progression. These are the major molecular mechanism of anti-cancer action of the flavonoids or other natural products. So we have performed some SRV and MTT assays on extracts, and in those extracts, we have found that ethyl acetate and certain aqueous extracts of Parthenium hysterophorus and T. cardofolia, they have provided very good efficacy, especially in case of Parthenium hysterophorus, you know, that is a noxious weed. So we have found more than 90% efficacy, even it is quite comparable to standard drug. this shows, as I discussed, why natural products? Because natural products have taken number of challenges for their evolution. And Parthenium hysterophorus being weed that has, taken a lot of good thing for its own benefit. That is why it is more destructive in nature, and it can just evolve itself for a longer time. So here we have seen the Piper longum also provides good cytotoxic action against different cell lines, MCF7 or DU145. These are the cytotoxic effect of the Bohnia variegata. We have also checked the mitochondrial membrane potential. So we have seen that few extracts, they have shown efficacy, not very good, because at the bottom line you will see camptothecin, a standard drug that is providing a good amyl depression of mitochondrial membrane potential in HL60 cells. But our product is also showing gradual improvement if we are going for concentration, and if we go for further purification, we are expecting better results. In cell cycle analysis, one of our extracts provided very good results. Here on top left, A hexane extract of P longum that accounted for 41% subG1 population as compared to camptothecin that was providing only 48%. So we got 41%. So these are the activities of natural products against some cancerous cell lines. So I'm coming to the end of my talk. So take home message from my lecture, my presentation is that just because of oxidative stress, we are getting a lot of problems in the living system, not only in human beings, maybe in all animal system, in all plant system. So especially for human beings or animals, natural products have enormous potential to act as antioxidant, hepatoprotectant, and anti-cancer agents. So with these words, I would like to just end my presentation. Thank you. Thank you very much. Now,
Sir,
yes.
sir, thank you, sir.
Yes.
Sir, that was a very informative session. So some questions are there for you. Shall I ask you?
Oh, sure.
So first question is, how to maintain the right levels of free radicals?
How to maintain?
Yes, how to maintain the right levels of free radicals?
Yes. See, we are currently living in such an atmosphere that more often we are exposed to some pollutants. So if we compare our lifestyle with our earlier days, means when there was less of pollution, I would just refer back to 2 months back period, when there was a complete lockdown. You might have heard the chirping of birds in your nearby. In the same way, atmospheric pollution level in Delhi has gone drastically down during April, May because of lockdown. Industrial activities were almost stopped at that time. So now why I am referring to it? Because if we consider the clean atmosphere, in clean atmosphere there is less likely to develop extra amount of free radicals. But if we are getting pollutants, obviously our genes are induced to produce, they are producing more amount of SOD, catalase, or maybe oxidative stress rectifying systems. They are inducing more to produce. So somehow we are making a balance, our body is trying. But at the same time, if this level goes again beyond the typical level, so our system cannot sustain, and we fall prey to it, and we just develop some disease or symptom like that. So obviously some lifestyle with natural food, we have, because nowadays we are taking a lot of medicines. You see, if minor headache is there, we are going for some analgesics or paracetamol. So our system is getting continuously overloaded. Even we talk about our foods, our food material, we are getting the spray of pesticides. A lot of foreign materials are coming in atmosphere. So a lot more buildup of xenobiotics is there in our system. So if we are somehow curtailing the pollution level, we can maintain our body system. So here, in normal circumstances, whatever some gurus they practice, that yogic exercises nowadays, maybe they provide some relief, at the same time pure food materials. You wash it properly, you try to remove pesticide, you go for scrapping external covering, etc. and with good food material. So that could be a strategy to counteract the excess buildup of free radicals.
Okay, sir. Another question is that compared to allopathy, how effective is the use of Ayurveda in suppressing free radicals?
You see, especially this is very interesting question. In the era of COVID, if you open the TV channels, lot more gurus they are coming and they are showing pranayam and all that type of activities, exercises. So obviously, if we talk about the allopathic versus Ayurvedic system, Ayurveda is nothing but the holistic system. You can say holistic means they are using total mixture of plant products. They are not going for pure products that we have talked about somewhere. In allopathic, we try to isolate pure products, maybe at microgram or nanogram level, that is highly effective. So sometimes we get synergistic effect, especially in Ayurveda system. If you see, you are in southern part of India. The climate in Kerala is almost uniform throughout the year. So there is a possibility for development of some specific kind of diseases in southern part as compared to northern part. Because in northern part, we are also getting cold climate and then typically warm climate. So depending on climatic conditions, different type of things are produced in different eco-climatic zones. So by taking spices, especially if we talk about some spices in controlled condition, too much of anything is harmful. If you talk about the basic principle of toxicology, you will find that too much of water is poisonous. Though water is not poison. Poisonous means it might create adverse effects in the living system. So we should take natural product in a limited amount. So we may get a better thing. And obviously, if you are falling prey to some disease that is not curable by this natural system, obviously one has to go for allopathic treatment because they are targeted drugs and they can produce rapid relief in time.
Okay, sir. Another question is, what's the effect of fasting on oxidational stress? Effect of fasting.
Fasting. Fasting. One is religious fasting. Other is people are taking fasting or making fasting as a practice in life to make them look slim that is there. So when we talk about fasting, obviously at metabolic level, stress is also there. You cannot say, because our system has been devised in such a way that we have full-grown intestinal system. So complete fasting, maybe without water, maybe we can sustain for 1 day or few. So fasting mixed with, I think, and this is my opinion, I'm not, so just because of fasting, you are depriving, for longer fasting, in longer fasting, you are depriving your body from essential elements. So after few hours, obviously, you need to take a small amount of nutrients so that your body is getting all the metabolic systems in normal standard way. That is my opinion.
Okay, sir. Sir, one more question is there. What is the role of vitamin E in the reaction? Role of vitamin E.
in the reaction means which reaction? Any specific or general role you are talking about?
I think specific role is there, but that student is asking what is the role of vitamin E in the reaction only.
Anyway, so as you know, vitamin E is a very good antioxidant. If we talk about antioxidant, vitamin C, vitamin E, even vitamin A, certain carotenes, they all provide antioxidant action. Now, why do we go for vitamin E? You see, vitamin E is a lipophilic compound. Tocopherols are lipid soluble. Vitamin C is water soluble. So our system is made up of lipoid material as well as aqueous material. So if we are taking certain amount of vitamin C, so whenever there is damage to lipoid membrane because of some ROS, reactive oxygen species, so this vitamin E has capability to scavenge radicals. It stops radical by providing them with hydrogen ion or electrons, and in turn it gets converted to a semi-quinone form of vitamin E. So whenever ascorbic acid reacts with vitamin C in the semi-quinone form, this gets activated and ascorbic is converted to ascorbyl radical. Again, that ascorbyl radical is converted to ascorbic. There is a cycle. There are 2-3 cycles, interacting cycles. They are continuously supporting each other. So that is why, especially in case of COVID, you will see that vitamin C is always given to all patients, because being water soluble, we cannot retain in the body. It can be flushed out of the system. A limited dose of vitamin E is also given whenever the patient requires. So vitamin E, vitamin C combination provides them stability to act as antioxidant. That is
Okay, sir. Okay, sir. Another last question is there. It's related to keto diet. They're
Yes.
asking what is the effect of free radicals on people having keto diets?
So most of your students are trying for dieting.
Yes, I think so.
So look, nowadays we also come across number of terms. Keto diet, sometimes 16 hours fasting, 8 hours feeding, number of things. Now, if you see our system, whatever the diet is there, what are the components in keto diet, maybe that is responsible for restricting calorie intake. So whenever there is calorie, if you talk about the calorie, I could not refer in my lecture, that calorie restriction is one of the strategies to increase the lifespan of any individual, be it human being or any. So limited small amount of food, lesser amount of food consumption may provide longevity. If you are talking about Rishi Muni, I mean, priests, those who are staying in the hills, Himalayas, all that. So they ate little of food. So their longevity was very light. So in the same way, what I think, so keto diet, there is a possibility of at metabolic level, to retain something which is responsible for normal metabolic machinery, but it will skip the excess fat and other things in the body. So one can get slimmer and slimmer.
Okay, sir. Thank you. Thank you, sir. Now I would like to express my gratitude towards you. All of us got a very detailed knowledge on oxidative stress and its complications. I express my sincere gratitude and thanks on behalf of our college, Department of Biotechnology and Microbiology, teachers, students, and other dignitaries. Thank you a lot, sir. Thank you.
Thank you, Dr. Saini, and thank you all members of GEMS family, your director, Dean Academic Research, all HODs, your principal, Dr. Naveen Mohan is sitting, I can see his smiling face. So I salute you all for this nice session. I hope the students will get good information throughout 10 days. So thank you so much.
Thank you, sir. Thank you for being with us. Thank you.
Okay, sir. Thank you.
Thank you.
Thank you.
Thank you. Say goodbye. And
Bye, sir.
My namaste to our Honorable Unni sir. I think he is not visible here.
Yeah, actually,
So,
he lost the connection, sir. He's trying to log in.
okay. So, my best regards to him.
Yeah,
We'll
sure,
keep
sir.
in touch. So, thank you all.
Thank you, sir.
Okay.
Thank you.
Bye. So, should I just go out?
കണ്ടേ ഈ